screen.
Good morning and welcome to today's Cannabis and Hemp Research Forum hosted by the UC Davis Cannabis and Hemp Research Center. I am co-director Li Tian. The purpose of our monthly forum series is to inform our campus community, members of the cannabis and hemp industry, and the general public on cannabis and hemp research being done at UC Davis, and also to provide an opportunity for guest external researchers to share their expertise. As today's presentation is underway, you will be able to post written questions using the Zoom Q&A feature. Our presenters will save time at the end to address your questions. This presentation is being recorded, and the recording will be made available in the next few days on the Cannabis and Hemp Research Center website at cannabis.ucdavis.edu under the Education tab. For today's research forum, we welcome two speakers, Dr. Patricia Holden from UC Santa Barbara and Dr. Sasha Nikolic from UC Davis. We will start with Dr. Holden's presentation. Dr. Holden is a professor of environmental microbiology in Brown School of Environmental Science and Management at UC Santa Barbara. She's also the director of the UCSB Natural Reserve System. Dr. Holden will introduce their project on investigating surface water emissions from cannabis cultivation sites. Dr. Holden, welcome.
Thank you, Lee. It's a privilege to be here, and I've shared my slides. It's my honor to represent a new project that's been funded by the Department of Cannabis Control today. The project is entitled, as seen here, Surface Water Emissions from Cannabis Cultivation Sites, Quantity, Quality, Toxicity, and Relationships to Farmers' Practices. You can see that I have a number of co-investigators from various institutions whose roles will be obvious as I go through the presentation. And then there are a number of researchers actively contributing to this project, which has been underway for a little bit over a year. And so a whole host of various institutions are involved, and we're very grateful to the DCC for support of this research. The motivation is that we have a lot to learn as a research community and in the agricultural cannabis industry of environmental dimensions of particularly legal cannabis cultivation. The graphic shown here is from a review article that members of our research team contributed to in 2021 that tried to assess what do we know about environmental dimensions and what's yet to be understood. There are different aspects depending on how cannabis is cultivated. The two upper frames showing what would be observed with a closed system, more energy inputs, more emissions, fewer emissions potentially, and use of municipal and other similar types of resources, and the use of urban land. Out of doors cultivation with probably what's more familiar in illegal cannabis grow settings in the lower right quadrant, which includes use of hazardous materials that are in some cases not permitted, and showing deleterious effects on water quality and wildlife. But we still have legal cannabis cultivation, of course, that's flourishing as per the current regimes in hoops and fully outdoors in various places in California and elsewhere. So we have a lot to be understood about the environmental dimensions of particularly these three quadrants of legal cannabis cultivation. So I want to just jump right into the project that, as I said, is underway. So I'm focusing more on telling you about the project today and how we're approaching it, and then showing you some very preliminary results. But we have 6 research questions that are driving this project that's taking place in Santa Barbara County. First, we have to understand where is cannabis cultivated relative to other crops, because to the degree that cannabis farms may be impacting surface water quality, biotic habitats, etc., it's happening within areas that are also intensively agriculturally important with conventional crops. We want to know what cannabis growers say they're doing with regards to chemical use in particular. We want to know what the plant products might be revealing. And we'd like to know, of course, what do we see in the chemistry and toxicity of surface waters and sediments from both a chemical analytical standpoint and from a standpoint of toxicity. And then taken together, what can we discern and recommend out of our findings? So why Santa Barbara County? Well, it turns out that Santa Barbara County is a really important region in the state for legal cannabis cultivation. In fact, as highlighted here, about 25% of all permits issued by the DCC on a county basis are to Santa Barbara County. And the cultivation of cannabis as a model region in Santa Barbara County is sort of widely distributed. We have all permits being distributed across the North and South County. And if you're familiar with Santa Barbara County, you would recognize the names of the cities and where they are located. It's not important here just to say that that we have, wide geographical distribution. And then in terms of the DCC permits, we have an important greenhouse growing region in Carpinteria, which is about half of all DCC permits, and then the rest are concentrated in mostly outdoor grows in the North County. So there's a lot of legal cannabis cultivation in Santa Barbara County. It's a busy industry here and growing industry. And also it has some interesting regional geographical distributions, including apparently, but that's yet to be understood, along growth formats. This map shows the Santa Barbara County Planning Department view of the permits that have been issued, just to show that they're distributed across the county with a concentration to the southeast in the city of Carpinteria, but widely distributed across the county. So our research has really three empirical data gathering axes. One, we're asking the farmers, what do you do? And recovering their reported practices, and tell you in a minute how we're doing that. And then second, we're sampling water and sediments in the vicinity of cannabis farms, but also conventional farms. And sampling leads to analyses both of chemicals and of microbial indicators of activities upstream. And then we're acquiring plant product, and through the work with a relationship with the California Department of Food and Agriculture, analyzing for product residues. At the nexus of these three axes is a spatially resolved environmental assessment, especially given all the intensive agriculture that's happening in our county. We do have a focus on pesticides, but we are looking for all types of chemicals that are associated with cannabis cultivation, including plant chemicals. But the focus on pesticides is interesting and important because the use and testing are not broadly standardized. And there's some recent literature that shows that the quantified residues are not uniformly compliant when you look broadly across geographical regions in the United States. And there are concerns expressed about product safety. So I'm going to just talk about these three axes and how we're going about this, and how much progress we've made so far. We're still in the early stages of this project, but it's coming along very nicely. So to discover the farmers' reported practices, first we needed to know who the farmers are, and there are three different databases from which we discover who's involved in farming. But our main inventory is from the Department of Cannabis Control permits, where we have the names and addresses of the growers. Then we developed an original questionnaire. We needed it to be concise and clear, something that could be administered conversationally, so that we could acquire information about chemical use, odor control, including chemicals used there. These are particularly important for the indoor grows in the South County. How is waste being managed? How is runoff being controlled? And any concerns that the farmers, their perceptions of the regulatory, climate, permitting, et cetera, that they would like to express, that helps us to understand why they're doing what they say they're doing. The questionnaire is an instrument and it needed to be beta tested. We did that internally and externally with regional, out of region growers, and refined. And then we invited participants to be interviewed through emailing, following up with phone and email, and reaching out through growers associations, and through a snowball sampling effect. We are expecting to be able to recover responses from a large fraction of our grower community, and we're hoping for that. So far we have been administering the questionnaire to a few growers, and we're very appreciative of that. All of the data is anonymized, and we are obligated and enthusiastic about absolute confidentiality, and this is required as a function of the Institutional Review Board permit to administer this survey. So we're very enthusiastic and excited about talking with growers, and happy that we've got this progress in having a really sound and efficient instrument to continue to administer. So then number two, so now, that's how we're finding out what farmers say they're doing. What does the environment tell us about what's going on? And again, this is both in the context of conventional crops and cannabis farms. So the first thing is that to understand what are the relevant data sets in the county, first regarding conventional crop farm locations relative to cannabis farms, a pesticide use in the county by conventional farms, water quality data that exists already. There's a Central Coast Ambient Monitoring, or CCAMP program. What are the sites? What are the historical data that might be useful as we start to interpret our data? And then what are the geographical features that we should attend to that might influence where we sample, including flowing surface waters, roads, and these types of aspects that would affect access. We map the farm locations, nominate sampling sites, perform field reconnaissance to determine which sampling sites are going to be feasible. And then importantly, developing a priori results analysis approaches, because we have, as you'll see in a minute, sort of a complex patchwork of different farms with cannabis farms interspersed in some cases. We have to anticipate how we're going to analyze and interpret our field analytical data in the context of what might be attributable to cannabis farms versus conventional farms. That allows us to finalize confidently site selection, and which we have done, and to schedule and perform field sampling, which is well underway, both in the North and South County, and to start analyzing samples, which is also underway by UCSB and San Diego State University. I'm showing this diagram here because our entry into legal cannabis cultivation and gathering all of the data that sets the stage for this field and laboratory and farmer interview-based research project requires that we really understand the institutional structures that can both help to guide us along the way. And we've had many questions, and we're very grateful to the many institutions who have been willing to engage with us as listed here, individuals. And so, but further, we needed data. And as our postdoc, Dr. Sanas Shamanara, put together here, many of the data sets were coming from local sources to put together a cannabis spatial layer, to put together a pesticide spatial layer. We're utilizing state sources, and some of them early state sources for the DCC licensee data set, et cetera. So many different data sources have been compiled, recruited, and determining where to acquire these, how to acquire them, and how to assemble them was actually a major job early on in this project. And we're really pleased that this has been done, and it sets the stage for a well-informed project and having selected sites for sampling moving forward. Just to say, the pesticide use reporting data for conventional crops is a really important data set that we've acquired, and we're very grateful for that from the DPR. And allows us to understand what chemicals are being used in what amounts in our county for different years, And in that vein, we understand how to interpret the data as our chemical analytical results are coming forward. That is, if there are pesticides that are reportedly used with particular crops in our region, and we're discovering residuals of those in our soil or water samples, then we're able to recognize that this is something that's actually being used in agriculture and make sense of the fact that it's there in the environment. The historical water quality data that we've recovered, this is just to say from the CDEN database, is available to us, has been acquired by the regional boards and their programs over different date ranges. And so as we start to interpret our data, we will refer to this to make appropriate comparisons, since it's important to reconcile our findings against history and against any current sampling and analysis that's occurring by the regional board. So just to show sort of part of the patchwork, and in this map of conventional crops in part of the North County, so this is just an example of a map that Dr. Shamanara has assembled, showing the patchwork of various conventional crops. And then over to the far right, a little symbol that looks like a teardrop with a white dot in the middle of it, and that's a cannabis farm. The blue dots are where there are the secant ambient aquatic monitoring sites historically dotted throughout. And so you can see that we have a very complex, in this example, North County site area around Santa Maria, complex landscape of different conventional crops, and then off to an edge, but still associated spatially with conventional crop areas, the cannabis farm. Another example is in the Carpinteria area, so in the South County, where we have this fairly high density, as you can see in the teardrop icons here, of cannabis farms. Again, they are within a patchwork of various other types of agricultural activities. And so it becomes very important that we have pre-thought how to sample our sites and how to choose our sampling sites such that we can analyze for attributable differences or attributable contributions from the various agricultural activities that are in spatial co-occurrence. We're doing this through what we call kind of a zone of influence analysis, where it's difficult to know precisely, and there's exactly how chemicals are distributing either through the air, through the water, or in other ways, other vectors from one location to another. But we have expertise in the team in chemical fate and transport sufficiently to recognize that there will be distribution. And so we've taken what we call a zone of influence approach that will be used in our analysis of chemical analytical results from our sampling sites. The red circle just defines either a 1,000-meter buffer here, for example, or a 500-meter buffer. And we quantify spatially for the farms that are now mapped to scale within those zones how much of the aerial coverage is cannabis farm versus non-cannabis. And we see in this particular example, there's about a 50-50 split with a little dominance of non-cannabis crops. In another example, we have zone of influence analysis indicating cannabis being a much smaller proportion of the aerial cropping versus non-cannabis. So this will be informative as we analyze our chemical results from the field sampling taking place in these locations. And then lastly, we do have sites where there is 100% cannabis, 100% non-cannabis cropping that's occurring. So those are important reference sites that should be devoid of markers of cannabis cultivation that we would find in the other sites to more or less proportional concentration to the agricultural areas. So this is just to say that we have pre-planned how we will try to allocate our chemical analytical results from the soil and sediment and water sample analyses from the various sampling sites. Okay, so this brought us to the point where we're able to confidently finalize our water and sediment sampling study design. We have locations that are selected. I haven't shown those here. They span a range of conventional crops and to conventional and cannabis mixed areas within that zone of influence as I described. We're collecting up to 108 water and sediment samples each over 1 year. We have 5 locations in Carpinteria and 6 in the North County that are sampled once per month for 9 months. And we're also sampling raw wastewater from the Carpinteria Sanitary District and flowing storm drains, as these urban locations will have these types of discharges as well that will carry important chemical signatures. So this is just showing how we're organizing our sample acquisition in the laboratory for water samples. We have samples that are being shipped to San Diego State University to the lab of Professor Unat Ho for non-targeted analysis, chemical screening, and also targeted analysis, and then also for bioassay and toxicity assessment by the lab of Professor Carrie Sant. Other water samples split on site are being pre-filtered typically, and we're assessing for evidence of fecal indicator bacteria, also microbial communities indicative of various activities on site that relate to all types of waste management. And then we're preserving filtrate for nutrient analysis, which will be indicative of potential fertilization effects on receiving waters downstream. We're making measurements in the field as we would routinely do by our experienced field sampling team. We're sampling sediments at the same time for also non-targeted chemical analysis and targeted analysis, and for preserving DNA for analysis of microbial communities. I just want to give an example of the methods and also some results from the bioassay measurements that are being made in the lab of Carrie Sant at, from San Diego State University. That is, in this particular method, which is a zebrafish embryo assay, which is a great representative for both aquatic life and also potential human effects. It's a monoorganism for both. Here, the fungicide boscalid, which is regulated in cultivated cannabis, is just being tested along with its metabolite M51OF1 in comparison to a negative carrier control, the solvent DMSO, at various concentrations to understand effects on embryo length, yolk sac area deformities. After a standardized exposure time and under standardized conditions. And the reason that we started with boscalid just to demonstrate our methods is because boscalid was discovered in a site that was adjacent to our sampling areas in Carpinteria, not necessarily emanating from any of our sampling sites, but we did know that it was in the region from having sampled and analyzed sediment previously. Professor Sant's lab has generated some interesting results to showing that boscalid is indeed actually fairly toxic along various lines of evidence with these zebrafish embryos in comparison to the control and also the metabolite. Reaching some conclusions about the teratogenicity of this chemical on zebrafish. So if we're finding it in the environment, and it is attributable to the agricultural activities, it will be of great interest and should show some evidence for concern that we would report. But again, this is example evidence that we can assess toxicity of a fungicide that's of concern in the industry, not necessarily that it's there, but we're prepared as we move forward with the analysis of the field samples that are actively coming our way. So in the plant, the third axis, the plant product residues, here we have a little bit of work to do. We're focused on acquiring product from regional dispensaries that are selling Santa Barbara material. We're still working on developing a legal strategy for doing that. We're very grateful for the consultation that we've been able to have with the University of California Office of the President, particularly Agnes Spalla, and also the UCSB Office of Research and the work that the DCC. We're very grateful for their counsel as well. And it may require scope modification to be able to proceed along a legal strategy for dispensary product acquisition for the research, hopefully working with dispensaries directly. We have a relationship through this project with the California Department of Food and Agriculture. They will be analyzing the dispensary material for residues, and they have a full pesticide panel that they'll be screening for and quantifying. This is enlarged way beyond the 66 regulated pesticides. Secondary analysis of their extracts will be performed by the lab of Professor Ho at San Diego State University. So in summary, what we have ongoing, and it's well underway, is a novel study of environmental influences of legal cannabis in Santa Barbara County, a very important region statewide for cannabis cultivation. We have these 3 integrated components, and we'll be looking towards the results to assess the alignment across reported farmers' practices, plant product residues, and water and sediment quality. This project is going to provide a good baseline understanding of environmental implications of legal cannabis in this region. And it will also result in recommendations that are regionally interesting, but also approaches to doing this sort of work at a regional scale that may be helpful across the state. We're very grateful to share this in-progress project, and hopefully you'll be able to, stay tuned and hear the final results as we move forward. We're acknowledging and very grateful again to the DCC for funding, also assistance from many state agencies as shown here, and the UCOP Policy Analysis and Coordination Office, as we described before. We're also extremely grateful to the participation and access by the Santa Barbara County cannabis growers, the individuals, and also growers associations. And we're really looking forward to working more with them throughout this project. And then individuals named here, Van Butzik, Barbara Harrell Hawthorne, Mark Batney, and I'm sure to have left off a few, have been extremely helpful in weighing in on this project early on and continuing to give us good advice and feedback as we navigate. Thank you so much.
Great, thank you so much, Dr. Holden. For audience of the webinar, please submit your questions through the Q&A feature of the Zoom. We can answer these questions at the end. Up next, Dr. Sasha Niklish will share their work on understanding the environmental impact of cannabis growth in California freshwater habitats. Dr. Niklish is an assistant professor in the Environmental Toxicology Department at UC Davis. Thank you for taking the time to share your work, Dr. Niklish.
Thank you so much. All right, let me just switch to this quickly here. All right, that was a great talk from Dr. Holden, and I hope I can, I will switch gears a little bit here, but I will refer to some of the slides that she was showing probably here and there. So I want to start with that slide. It is the title of today's talk, Environmental Impacts of Cannabis Growth in California Freshwater Habitats. And it was actually the title of a grant that we submitted also to, back then, the Bureau of Cannabis Control in 2020. And it unfortunately didn't get funded, but it's not a problem. I'll just, you know, like talk through these ideas that we have, because we're actually also about to resubmit that. All right. Let me just get you. All right. So I just want to start out like 1 minute, just like telling you who we are. I just joined this Cannabis and Hemp Center here at UC Davis this year. So maybe it's good to know what are we doing and how are we connected with our topics, research topics to the center. So I'm a protein biochemist in background. We have a small lab that you can see here Currently, we have 2 graduate students, a couple of undergrads, and you can see by the framing of the pictures what they're working on. We have 3 general topics in our lab. So protein biochemistry is number 1, I would say, like this is the main focus. So we're working with transport proteins, and I will talk a little bit about this if we have time later. Drug transporters that determine chemical uptake in organisms. We work in mice and fish and in honeybees. We also do analytical chemistry, so we're trying to understand levels of chemicals and other compounds in food organisms. And we have a small interest or small subgroup that is interested in trying to predict accumulation of chemicals in organisms. Okay, so this is our lab, and so with this background, with this knowledge, we try to apply this actually also to working with cannabis or cannabinoids. And this is what I would like to talk about today. So this is a 2020 grant that we submitted, as I mentioned earlier, and which did not get funded so far, but I would like to discuss a little bit what was the general idea here. And as you can see here, I pulled together, back then, I was lucky to pull together like this team of experts working in aquatic toxicology, analytical chemistry, also in risk assessment, and histopathology, for example, to help me to fulfill the requirements for this grant and also for these grant ideas. So this is the main part that I would like to talk about today. And then as an icing on the cake, if we have enough time at the end, I would like to also talk about the grant that I'm currently submitting, actually in 2 weeks, to the Department of Cannabis Control, which is more on the chemistry, toxicology, and the brain disposition of e-liquids, so from vaping, okay? So I'm working there with another set of colleagues together on this. Let's first talk about this grant and the grant idea that we had back in 2020. We were interested, background, of course, here in the Department of Environmental Toxicology is trying to understand how chemicals and cannabinoids, for example, also like natural compounds, could affect like organisms, humans. In our case, we were interested in California fish and also in the juvenile fish, so in a more vulnerable stage. So the juvenile fish in rivers, Sacramento River, for example, that would face runoffs not only from agricultural operations, but also from wastewater treatment plants. So we had it, our team was kind of divided into like 3 subgroups. We had people working on the exposure level. So we're trying to understand what cannabinoids do we actually detect in wastewater treatment plants and in the effluents and the surface waters, of course. And what degradation products and what biotransformations are happening in the wastewater treatment plant, but also then what can we detect downstream in the surface waters. And then a second team was trying to analyze how do these compounds, these degradation products, these metabolites affect the benthic community. I'll talk a little bit more about this using nematodes. So as a first step, we used nematode as a benthic community, but also invertebrates to see if there are any toxic effects or changes in the composition of the trophic structure in these communities. And then a final stage, of course, where I was mostly involved, and I will talk about this part more, is the toxicity. Can we detect any toxicities of these compounds, these cannabinoids that we're detecting in the surface water on California fish? Is there a behavioral effect? And also, do we see any pathologies in their tissues? Okay, so the main questions that we had is like, what's the stability, bioavailability, environmental fate of these cannabinoids and that come out of the wastewater treatment plant? So they are, in fact, cannabinoids that cannot get filtered out still until today. And they end up in our surface water. So we were interested in like what's their environmental fate, and then trying to understand what effects would that have on nematodes, on invertebrates, small invertebrates that could serve as food, of course, along the trophic chain for California fish in these river streams. Okay. So what do we know about these exposure, bioaccumulation, and toxicity that I showed you in these three subparts that we were trying to analyze to perform actually a risk assessment for these cannabinoids. So as I mentioned, for the exposure part, little is known, but we have a couple of reviews here. You can see here from Apul et al. 2020. It is like a review of global sewage waters, tap waters, drinking waters, and surface waters. And you can see there's ranges, of course, that have been detected for certain cannabinoids, mainly the arguably inactive, like psychological or neurologically inactive metabolite, the carboxylated THC, right? And so most of the data we have actually about the levels of this compound, this metabolite, that comes out of the effluent of the wastewater treatment plant, but also CBD, as you can see. There's unfortunately like very little known about these levels of these metabolites and other metabolites in surface waters, as you can see here. But then we detect and we monitor also, of course, much more our drinking water, so we can detect some of these compounds in our drinking water and also in our tap waters, right? So if you look at these different metabolites, as I mentioned, most of these monitoring data that we have is on this carboxylated THC cannabinoid. And here, how it out, for example, like 2 years ago, could show that in some of the effluents of the wastewater treatment plants, this is actually in New York State, they detected THC, the carboxylated metabolite, at 772 nanograms per liter in this effluent. I also write down in parentheses here, it's roughly like 2 nanomolar. And I'll talk a little bit more about this maybe later, because as a biochemist, I'm interested in the molarity and concentration of these compounds that could act on proteins, right? So 772 nanograms per liter. This was actually, I think it was like measured a week after Thanksgiving. Yeah, go figure. And these were relatively high levels. So the range is, of course, like as you can see here, starting from 6 nanograms per liter to 772 grams per liter. So we know a little bit about some metabolites of these cannabinoids that we find in our surface waters or in the effluent of the wastewater treatment. So what about the bioaccumulation that we're also interested in? So the first thing that we did, of course, was ask NCBI, our PubMed, our database on what is published. And I actually did this again yesterday, and you can see there's not a lot out there about bioaccumulation. So when I talk about bioaccumulation here, I mean the sum of accumulation of these cannabinoids in any organism, by the way. So I didn't limit it to fish here. So any organism, I just wanted to know how much of these cannabinoids can bioaccumulate, can concentrate, can go up the chain, like the trophic chain, so magnify. And I use this, of course, like different combination of these words, but there's not a lot of information, of course, still, right? December 7th, that was yesterday when I prepared the talk, not a lot of data on this bioaccumulation and trophic transfer in any organism. So we still think, of course, there's a gap of knowledge here. And research that needs to be performed, understanding how they could maybe transfer from food organism along this trophic chain. And then finally, of course, like what do we know about the toxic effects of these cannabinoids that are detected in these effluents of the wastewater treatment plants? And now I'm referring back to what Dr. Holden was saying, like most of it is actually, of course, known about our model organism, the zebrafish. So you can see here, if you type in PubMed, also yesterday, what data has been published. There are a couple of studies out there. If you look at them carefully, you can see there's a lot about developmental effects of these cannabinoids, specific cannabinoids, CBDs, THCs, of course, and also their effects on swim performance, for example, and metabolic pathways of these zebrafish. And that is actually something that we would like to exploit or wanted to exploit with this grant, also like trying to use their swim performance, their photo locomotion to determine the neurological effects of these cannabinoid metabolites that we detect on fish larvae or developing fish. Okay, so that is, of course, that is so far what was known. And then finally, of course, the toxicity part that we were mainly involved with. We were lucky to collaborate with Dr. Richard Connon and Dr. Emily Segara, who have access to two species like fish here in the native fish from California, and that we find in our rivers. One of them was the Sacramento sucker. So here we try to use this fish as a representative fish for lake and river bottom feeder fish, right? So you can see here an adult fish as an example, but we would also have access, of course, access to the larvae and the eggs of these fish. And then as a second fish, we would take the rainbow trout, a very well characterized model organism also in the aquatic toxicology fields. And using that as a migratory fish species that we know it will be present along the rivers, the Sacramento River as well, from the source to the end of this river. So we had these two model species that we would like to target and develop the assays for juvenile fish exposures. And similar to what Dr. Holden was also showing earlier, there is, of course, like assays to assess toxicities of these compounds of pesticides, and we'll use the same assays and endpoints, of course, to assess the effects of these metabolites of cannabinoids. So mortality, of course, is one. The morphometrics, so looking at how does these fish larvae develop? Are there any reduced development of the yolk sac, for example, the size of the yolk sac, or is the development of any fins interrupted or impeded during this development upon exposure, of course. And then also understanding the body burden. So BB stands for body burden here. We try to understand how do these, if and to what extent these metabolites can accumulate in these juvenile fish. And as we all know, this yolk sac is a very lipid-rich, lipophilic, hydrophobic environment that is almost like a sponge for many of these compounds, pesticides that are hydrophobic, but also for cannabinoids that are hydrophobic, right? So we try to understand and all that, like what goes into these fish, how much is it, and how could that affect the development of this fish. And then in the second point, we would expose developing larvae, but also late stage larvae or adult fish to these cannabinoids and metabolites, and trying to use these observational, these behavioral endpoints upon exposure to assess the effects on neurotoxicity of these cannabinoids. So here you can see on the left side in the Connon lab, they have these nice, it's called the Daniel Vision. It's an observational chamber with a camera that allows you to observe different types of mobilities, motions of these fish in a 96 well plate or 12 well plate, you name it, upon exposure to different amounts of your compound of interest, right? So you can see here, like there's an increase in bursting and the velocity of freezing. The fish doesn't move at all, or it's just cruising. So these are common patterns that are referred to a vehicle control, of course, upon exposure to different types of chemicals, pesticides included. But we would like to, wanted to use this also to test what are the effects of these cannabinoids, right? Like so on these fish. So if we know that they get into these fish, do they have any neurological effects that could affect the swimming performance of these juvenile fish? Swimming performance, of course, is very important if you would like to escape from predators, especially during your early development of such fish that are important for this California. So that was the toxicity part. So I'm showing you pretty much like today only this very small part here you can see of what we were interested in. And as I said, we're of course, we would like to resubmit this idea because as of yesterday, it seems there's still research needed in this field. And hopefully I can pull these people back together on the table, these collaborators, so we can do these nice studies on the risk assessment of cannabinoids and their metabolites. So as I mentioned, this all idea, no pun intended, went up in smoke, but we would like to, right, like never give up, that's what everyone tells you, resubmit this grant probably in the next cycle, not in this current cycle, because this is really one of the main interests that we have in this work. So I think I have like 5 minutes left, 5 to 10 minutes left probably, and I would like to talk a little bit about briefly about what we're up to now in the current submission phase of the Department of Cannabis Control. So I'm working here with Tran Nguyen, Dr. Tran Nguyen and Dr. Ken Pinkerton, like an aerosol expert, Tran, and Ken Pinkerton, an immunologist and he works on cell toxicities and immunotoxicities in our department, and we're trying to understand what compounds, what metabolites are actually built during the vaping process. Yeah? So if we vape different e-liquids with synthetic or natural cannabinoids in them, are there any toxic degradation products that can develop? And how does that possibly affect the neurological effects of these cannabinoids and their degradation products once they are taken up into our brains? Okay. So this is just like showing you like the landscape of what is out there of cannabinoids, metabolites, and other compounds actually that could be in these vaping liquids and these e-liquids. So we have regular cannabinoids on the top left, as you can see, and they're metabolites that are typically produced in our body by the liver, for example, that would, like be able through the bloodstream to pass into the brain, to get transferred into the brain. We also have in these e-liquids very often flavorants and modifiers, as you can see here. So we know a lot about that as well. We have synthetic cannabinoids and their metabolites that are out on the market. And very recently, we also know that there are metabolites, sometimes toxic metabolites, formed during the vaping process, mainly due to the combustion temperatures that are reached during the vaping process, right? So there's a lot of chemicals out there, and we try to understand, okay, so these are all in our bloodstream. They could possibly be in our bloodstream. So which of them, how, accumulate in the brain? Can they accumulate in the brain, cause toxicity in the brain, or maybe synergistic effects, like effects that we are not planning on having on our receptors that confer these effects of cannabinoids. Okay, so this is pretty much like what we are trying to understand. Like Dr. Nguyen wants to identify the level, identify the compounds, the metabolites, and also the levels in these e-cigarette liquids. I would like to be able to predict if these compounds, these metabolites, can actually transfer into the brain. And then we would like to also assess the toxicity, the cellular toxicity and immunotoxicity, Dr. Pinkerton's lab as well. And I only have 2 more slides where I might have to torture you with as a biochemist. So how do I want to analyze, like, if these compounds, if these metabolites, these novel cannabinoids, as we often refer to them, if they can pass into our brain? So you can see here in all of our biological barriers, including the brain, the liver, the intestine, the kidney, for example, we have so-called drug transporters. We have transport proteins that regulate chemical uptake, right? So they are protecting, if we focus here on the brain, some are pumping chemicals or compounds and nutrients into our brain. And then these 2 guys here, and I specifically am interested in these 2, PGP and BCRP, they are protectors of our brain. They're pumping out drugs and chemicals that don't belong into the brain. So they're kind of like, we often call them molecular vacuums, because they're getting rid of toxic metabolites and keeping our brain clean from these metabolites, but also any other type of drug or chemical that doesn't belong in there. And we often also refer to them as the bounces of the cell. So here you can see a structure of one of these proteins that typically, when a drug wants to pass through the blood-brain barrier here, this is a membrane, these transporters will recognize it and pump it back out. So for some of, referring this to our cannabinoids, for some of these compounds, the THC, for example, is a substrate for this transporter. It recognizes it and immediately pumps it back out into the bloodstream. So there's kind of like this homeostasis, right? Like, so these are transporters that regulate the dose, the final dose that gets into our brain, right? So that some is known, some of these compounds, we know how to interact with these transporters. In fact, in drug development, the FDA has a recommendation that all new developed drugs first have to get tested against these 2 transporters in particular before they can get on the market, because they are determining like how much of a drug can get, for example, into your brain to treat Alzheimer's or other neurological illnesses or diseases, right? So you have to, these transporters are highly efficient, and you have to understand how these compounds interact with these transporters. And how does that relate to our cannabinoids? Well, some of these cannabinoids, we have data, recent data, or data that is like over 10 years, 15 years ago, have shown that some of these cannabinoids can actually block these transporters that usually keep your brain clean from toxic metabolites. So depending on the concentration and the type of cannabinoid, some of these transporters in our brain can be inhibited by these transporters. And here's the other transporter, ABCG2 or BCRP, is also inhibited by these cannabinoids. So this is something that we're really interested in. Okay, there's a knowledge about some of these cannabinoids can inhibit these transporters. What about these new vaping cannabinoids? Like, do they inhibit? Can they activate these transporters? And how does that overall, like depending on what you are ingesting, what you're consuming, how does that affect or how does that impact the neurological effects of these cannabinoids? And this is pretty much like the idea that we have, depending, of course, of the composition of your cannabinoids in your strains that you're consuming. The ratios, we know they're important for the effects of THC on your brain, of CBD to THC, but also the mode of consumption that I mentioned earlier, vaping, smoking, or edibles will produce different types of metabolites. And we know that they are converging at these molecular targets of transporters in your brain barrier. So understanding how these compounds interact with these transporters will then allow us to predict what compounds can be eliminated, but also what compounds will be absorbed into the brain. And this is the idea, the overall idea, trying to understand, are there any metabolites that don't necessarily have to be directly toxic to the humans itself, but they could block these transporters. And what it means is they are causing an accumulation of THC, too much THC, into our brain. So overstimulation could cause, depending on if you're vaping, as I mentioned, or eating or smoking, you can have an overstimulation of THC. Or do they inhibit these transporters and other compounds that usually get eliminated by these vacuums, accumulate in your brain and cause more toxicity? And finally, of course, we want to understand other compounds that maybe can upregulate these transporters, and too much THC leaves your brain in a very quick or brief moment and time interval, which could enhance addiction, right? So if you have like THC flush, but you have also like a depletion, a very quick depletion, this could increase the addiction to cannabinoids in this case. And this is something that we're really highly interested in this new grant. And that's pretty much it for it. And I hope if anyone has questions, I would love to answer them.
Great, yeah. So please submit your questions in Q&A. I will start with one for Sasha, then another one for Trish. Okay, Sasha, you talk about this modeling approach to predict whether some of the vaping products may, have some effect in for the blood-membrane barrier. I'm a plant biologist, I have to tell you first, so my question might be naive. So, can you, probably people do this already, like reconstruct like an in vitro system, right, with the membrane and the transporters and test this product that way. So probably it's not as, well, it's not a human system, but at least can, provide some experimental evidence for the inhibition.
Yes, exactly. That's what we are trying to do. So before we would go into a human studies in general is far away. But before we would go into like an in vivo model, we have an opportunity here to use like key predictors, which are these transporters. So as I mentioned, like they can determine up to 70, 80, 90%, or kick out, eliminate 70, 80, 90% of a drug or cannabinoid that would usually get into your brain. So we can actually produce these proteins in vitro, in a detergent, in a solution, and we can measure them in 96 well plates, which means I can do literally 96 reactions with compounds within 15 minutes to measure what combinations, what ratios, what I was talking about, what combinations of metabolites that we are hopefully detecting then with in these aerosols, for example, what combinations are causing inhibition, what combinations are causing activation of these transporters, and the system of measuring the interaction with these transporters has already been shown for drugs. So we know that if a drug inhibits this transporter in vitro in my little dish, that is actually also a drug that accumulates in humans. So we are just using the pharmacological knowledge, using a high throughput or medium throughput assay to screen combinations and other novel cannabinoids then in vitro first, before we would ever go at a later stage, of course, in vitro. Yes.
Great, thank you. Oh, okay, I have a very quick question for Trish. So I'm very interested in the survey approach. So what was kind of the response rate like from the growers when you reach out to them? Because this is a very useful tool.
Yeah, thank you for asking, and it's forthcoming. So far, we've had several responses and the experience has been really good. Sanaz has been performing these interviews and we're very grateful to those who participated and we're expecting this snowball effect. All three of the respondents have agreed to share the good word, that it's a good experience and that they have the chance to have a voice and to express not only what they do, but their opinions and their views on the environment here and the context in which they grow. Thank you.
Great, thank you. Yeah, we have 12 o'clock already. So thank you, Trish and Sasha for the wonderful presentations. I really enjoyed learning about your work, and also thanks to everyone for joining us today. I hope you all have a wonderful winter break and see you again when our forum series returns in January. Happy holidays, everyone. Bye-bye.
Happy holidays. Thank you.
Thank you.